Good afternoon to everybody. Well, I'm so pleased to be here to introduce Dr. Adrien Guillot. Adrien Guillot was born in Lyon, France, but then he moved to the outskirts of Paris, where he's trained as a biologist, as a PhD, and then spent 4 years in the NIH working. Now he moved to Berlin, but I'm going to be more formal, and before giving the floor to Adrien, I'm going to read the highlights of his very interesting career. He's a research group leader at the Charité University Medical Hospital in Berlin, in the Department of Hepatology and Gastroenterology, led by Professor Frank Take. He received his PhD at the University Paris Iscritil, where he studied the roles of interleukin-17 in liver pathogenesis and liver regeneration through ductal reaction. He's going to discuss the ductal reaction this afternoon, which is thrilling for me at least, and I hope for most of the audience, under the supervision of Professor Fouad Lavdil in the interim team. He pursued with a 4-year postdoctoral experience at the National Institute of Health, where he conducted 2 projects related to targeting liver acetaldehyde dehydrogenase 2 in alcohol use disorder and monocyte macrophage response to acute biliary epithelial cell injury, and then joined the Professor Take laboratory as a postdoctoral fellow with the support of the Alexander von Humboldt Foundation grant. And his international research group focuses on dissecting the intricate cell-cell interactions involved in liver disease progression, with a particular focus on the events taking place in the liver portal area. Alongside Dr. Guillot is optimizing methods related to multiplex immunofluorescence and digital image analysis, as well as liver-on-a-chip multicellular models. He's a member of the editorial boards of JF Reports and eGastroenterology, and reviewer for leading journals in the field, and is also active in science advocacy at the European level. So thank you, Adrien, for being here, and we really appreciate having him here because he's had some family troubles that require his immediate attention, but in spite of that, he had decided to bear with us at least until tomorrow. So thank you again, Adrien, and the floor is yours.
All right, thank you very much for the kind introduction and of course for the invitation. It's my great pleasure to be here in Barcelona. So today I would like to tell you about our published work, but I will bring you through a red line, which I hope you will recognize as pretty important in liver diseases. So the title of the talk today is Cellular Interactions in Liver Injuries and Regeneration, focus on the portal area, which is quite a broad title, but you will see why I made it so broad. And the subtitle would be Insights into Multiplexed Research. So I will not offend you through going through the details of this schematic, which is the conventional progression of chronic liver diseases. But I would like to make some points on this. Actually, most people, when they show you this progression of liver disease, they are going to speak about research that focuses on hepatocytes, immune cells, and fibrogenic cells. But actually, there are other cells in the liver, and for example, cholangiocytes, so the biliary epithelial cells lining the bile ducts. And it's actually not very well known what those cells would be doing in liver diseases. And as well, we could ask how much do we actually know about what is happening in the portal area repair. And now I will start my talk by showing you my conclusion, just to make sure you get the key message straight. So cholangiocytes are not bystanders. And macrophages in general, but it's the case for liver macrophages, are very diverse immune cell populations. And just on this scheme, you will understand why we decided to put the cholangiocytes really in the middle of a complex, intricate cellular crosstalk network in the liver. So let's speak about cholangiocytes, or biliary epithelial cells. Just a few points. So they are epithelial cells. They line the intrahepatic and extrahepatic bile ducts. They are themselves very heterogeneous in terms of size and functions, but they globally contribute to the bile composition and the bile flow. They actively participate in liver repair mechanisms, and this is what we would call as part of a ductular reaction. And the role of the cholangiocytes in innate and adaptive immune responses is emerging in a couple of studies recently. And when we speak about liver macrophages, I like to call them a modern family. So they are, again, a very, very heterogeneous cell population with very dynamic features as well. So for instance, it has been the case that for quite some time we were speaking about M1, M2 macrophages. Now we advocate for moving away from this. Like it's really a much broader range of activation phenotypes in the liver macrophages. And there have been events of macrophages switching from one phenotype to another one. And then comes the question, what are the specific features of those monocytes, macrophages in steatotic and cholestatic liver diseases? So this is what I will focus on today. And also, how do these macrophages spatially interact with their microenvironment? And before we get into that, I will make a short technical note here. When I speak about multiplex research, so that actually led us to develop a specific protocol for multiplex immunofluorescence, which I will introduce to you now, in case you are not familiar with this. So in general, most people would know the advantages of performing immunohistochemistry on FFP tissue sections, so formalin-fixed, paraffin-embedded. We have access to large collections of archival samples. It's pretty simple storage and shipping of those samples. And the tissue and cell morphology is generally well preserved. Now, the advantages of moving towards multiplexing in those samples, it's clearly we have an overview of the immune and parenchymal cell numbers, location. We can contextualize where those cells are they in tumor region, healthy regions, portal area, and so on. And at least the method we chose to use in our lab is we consider it as an accessible method, meaning we don't need a specific microscope. We just use a fluorescence microscope. And we don't use commercial kits. We use off-the-shelf kits that should be available in most labs. And by doing this, we can really visualize. So we can perform cross-staining of multiple markers. I put a number 15. Theoretically, there is no maximum. I mean, of course, you can imagine there is some physical damage at some point. And we can visualize the cell distribution through the sample. But for us, the most interesting part is we can quantify. So the goal is really to extract the numbers from those images, from these complex images. And this is just an overview showing you that indeed there are quite complex cellular landscapes, particularly in the portal area in liver disease. So how do we perform multiplex immunofluorescence in our lab? So it's pretty much a conventional immunostaining protocol. You would recognize the steps here if you are familiar with this. The main difference is repeat X times those steps here, in which we perform antigen retrieval, immunostaining, imaging, and then chemical antibody stripping. So that means we just remove the antibodies after taking the images, and we perform a next round of imaging. And this is just an example of the types of images we can generate from one mouse liver in this case. So here we show you a DEN plus CCL4 mouse liver, so highly fibrotic and inflammatory. In this sample, we performed 7 immunostaining cycles for a total of 13 primary antibodies plus DAPI and Masson-Stra chromin variant. So that's just illustrating you what kind of data we can generate. And then if we speak about data analysis, I will not detail it today, but I really want to emphasize, do not underestimate the complexity of this step. So it does require a lot of time, a lot of expertise to extract the numbers from those images. And in our group, I really like to focus on freeware. So we don't use commercial software, we use freeware. We develop our own plugins as well for some parts of the processing of images. And in the end, you would see what it looks like here. So for example, this is the raw image. I mean, it has already been processed because we already merged a couple of markers together. And next to it, you would see the result of a segmentation of this image. So this is just showing you the different cell types we identify thanks to the immunostaining we performed in this tissue area. So this is a protocol we have been working on intensively for the past 5 years, so to say. We have been pretty successful, honestly, in publishing it, and these are just some of our collaborations that we could run through this protocol. And for the next parts of my talk, I will focus on the publications that I highlighted in bold here. So now that I introduced you the technique, let's see how we applied this technique to this project here, in which we wanted to apply multiplex immunofluorescence to human liver diseases. And I will focus on those two chronic liver diseases, PSC and MASLD. So PSC for primary sclerosing cholangitis, it's a conventional cholangiopathy, let's say. And MASLD, metabolic-associated fatty liver disease, which you must be aware of. It might sound a bit surprising why we decided to focus on those two diseases together. The motivation behind that was these are two chronic liver diseases, slow progression with increasing stages of inflammation and fibrosis, and importantly, they are both characterized by intense ductural reaction. So what we did in this project was to collect samples, so tissue sections of liver from those patients at different stages of MASLD, MASH, or PSC. We performed sequential multiplex immunostaining on those samples for a total of between 10 and 20 markers. That depends on the batch of staining. In the end, we had almost 100 human tissue sections and 35 mouse liver sections. So that shows you the protocol we use is rather cheap because we can apply it to multiple samples. And you will see across the slides, the human MASLD samples were grouped by fibrosis stage, F1 to F4. And this is showing you the markers I will mention today, which were part of the panel and applied to all those samples, at least the human ones. CK19 for cytokeratin 19 to visualize the biliary epithelial cells, EPAR1 for hepatocytes, IBA1, CD1, 63, and 16 for monocyte macrophages, MPO neutrophils, and CD3 for T cells, and alpha SMA for fibroblasts. So this is just showing you some typical images we generated in our sample cohort. So you see the scale bar. You can appreciate we really try to image very large areas. This left sample is a healthy human liver, so it's pretty much blue. That's stained for EPAR1, the marker of hepatocytes. So the healthy liver is filled with hepatocytes, and we really have to zoom in this tiny area to find a portal area with the yellow cells here marked for CK19. So these are healthy biliary cells. And now if we look into MASH fibrosis stage 4 or PSC samples, so these are really end-stage liver disease, you see the blue areas are shrinking, so this reflects the progression of a disease. And if we zoom in those fibrotic and inflammatory areas here, we can really see this extensive ductural reaction, meaning the accumulation of CK19 positive cells. Oops, sorry, you already saw that. So the first thing we did was just to look at where the cells are located. So here you have the same samples, and these are density maps. So it's not staining intensity. It just shows you where are the cells located on the image. So if it's red, it means there is a high density of cells. So total cells means DAPI. We just look at where the cells tend to accumulate. The healthy liver is pretty homogeneous, again, but the diseased livers, you see these very densely populated areas, again reflecting the inflammation and ongoing fibrosis. The hepatocyte staining, it's the opposite, tends to decrease. And we have a massive increase of CK19 positive cells, again, those ductular cells, as well as IBA1 positive cells, which we use as a marker for whole monocyte macrophages, MPO neutrophils, and CD3 for T cells. Now, I told you the goal for us is to extract numbers. So what we did here first was to characterize the cohort. And we could see, along with increasing disease stages, in MASLD, there is a decrease in the EPAR1 positive area. So the hepatocyte positive area is shrinking. On the other hand, the positive area for alpha SMA is increasing. That's the ongoing fibrogenesis. And again, this increase, gradual increase in CK19 positive cells. When we look at immune cells now, it's kind of the same picture. So we do see an increase of IBA1 positive, again, monocyte macrophages, neutrophils, and T cells. You might appreciate the MASLD is pretty much strongly associated with myeloid cells, whereas the PSC tends to have more of the T cells. You might appreciate, so these green points here, one point is one patient sample, and the PSC cohort was pretty heterogeneous in this case, so we just grouped that all together. Now, for the next stage, which is, I would say, the most interesting part of this study was to really draw a landscape of the macrophage populations in those samples. So what you see here, it's again those 3 typical samples that we selected here. And each pixel on those maps, those plots here, represents 1 macrophage. And then the color of the plot represents the staining intensity for the different markers shown here. So IBA1, you see it's pretty much the same color everywhere, so the expression is homogeneous through the whole liver. So we use that as a macrophage marker for all kinds of monocytes and macrophages. But if we look at CD16 and CD163, there is a drastic difference in where the cells would accumulate. So the green dots here are the CD16 high positive macrophages. We assume remaining Kupffer cells. And the blue dots would be the CD16 low monocyte-derived macrophages in this case. And they really accumulate in this perinodal area, perilobular areas here. And that was the same in MASH and PSC. When we quantified that, so 1 dot is 1 patient from our cohort. And again, we could really spot that the macrophages that tended to be in the EPA1 negative area had much lower expression of CD16 and CD163, which we assume is related to a loss of scavenging functions, because those are markers related to the scavenging functions of macrophages. And what's more, we could really spot that the distance between those macrophages and CK19 positive cells was a highly relevant parameter in these cohorts of patients. So what you see here, these IBA1 positive cells, so the macrophages, tended to be the ones being the closest to CK19 cells compared to T cells and neutrophils at least. And they also tended to be the ones in the highest number in a short distance from the CK19 positive cells. And what's more, in this graph, the blue bars here reflect the numbers of macrophages being far away from CK19 positive cells. And you appreciate this number is not changing much between the different disease stages. But what is drastically increasing is the number of macrophages being close to the CK19 positive cells. And those macrophages that are close to the CK19 positive cells are especially the ones, again, having low expression of CD16 and CD163. So again, 1 patient is 1 dot on these graphs, and we could establish that as a significant finding in the whole cohort. I'm trying to keep it rather simple. If you wish, we could go through more complex discussions. So we could spend a few hours looking at this. This is just a matrix compiling all the imaging parameters that we correlated with the clinical parameters we had on those patients. So of course, we spent some time looking at this ourselves, but we also just fed a machine learning algorithm with this data and basically asked through a random forest algorithm, what is the typical signature of parameters, imaging parameters, that would associate with disease progression in those patients? And quite interestingly, one of the parameters that really popped up in this analysis was the number of macrophages being close to CK19 positive cells. And you might realize there is not much related to T cells or neutrophils in this cohort. And then we could generate some kind of formula that we applied to a validation cohort, and we could again establish this relation between CK19 positive and IBA1 positive neighboring accumulation and disease progression. What was more astonishing, so far I showed you muscle D and PSC only, but we had the same patterns in PBC patients, primary biliary cholangitis, so another cholangiopathy, and in severe alcohol-associated hepatitis, which is, again, another type of chronic liver disease. So in the end, if I conclude on this first part, we could establish that the monocytic macrophage accumulation is a common hallmark of chronic liver disease progression across very diverse etiologies. And this imaging cytometry that we performed, so really digging into images and isolating the phenotype of singular cells, pointed out that IBA1 positive CD16 low, CD163 low macrophage accumulation near CK19 positive cells marks chronic liver disease progression. So this work has been published in Hepatology, and just to mention, because it's a sponsor, it was a collaboration with Gilead, so they will be happy. I mentioned their names. And I will now go to my next part of the talk. So now that we showed cholangiocytes and macrophages tend to be stuck together during disease progression, of course, arises the question, are they actually both involved in this progression? So now I'm rewinding a bit back in my research journey. So when I was a postdoc at NIH, there was this question that there was no appropriate model to study cholangiopathies, at least in mice. So the current models, most people would be using biliary injection, MDR2 knockout mice, bile duct ligation, or diet-based injuries. But each of these models has, of course, limitations. No model is perfect. They would induce chronic injury, massive necrosis, non-cell specific injury, or have extrahepatic consequences. So the goal was in this project to establish a conditional, acute, and cell-specific model leading to biliary cell death and regeneration. So that was actually before the work I showed you. And I will spare you the details of the model. Just please remember, this is really a model of acute targeted biliary cell death using transgenic mice. And this is what I show you here. So within 3 hours after we inject the toxin inducing this model, we have very localized tunnel staining in the portal areas, precisely in the biliary epithelial cells. And this staining is gone after 48 hours. So this is what we call acute injury. And it's reflected here as well. You have a kinetic of ALT, total bilirubin, as well as gene expression of FXR and CYP7A1, which show you that there is really a peak of cholestasis-associated response, at least, around between 3 and 9 hours. So this is what we call the acute model, coming back to normal after 24, 48 hours. So we were very happy. That seemed to be a model to study really these acute events following biliary cell injury in a rather clean way. And then we decided to look into the events following this acute injury. So one thing that might be relevant for some of you here, we could spot that there was just by this very targeted biliary cell injury, this was enough to affect the liver blood microcirculation. So we could see a decrease in the blood circulation with the flux, and also having an increase in the portal pressure in these mice. So portal vein pressure, just again by this very clean acute biliary cell injury. And I told you it's a reversible model. So we did see a peak of repair mechanisms, so proliferation of the remaining biliary cells happening at 48 hours. So there is ability to regenerate or repair the biliary tree in a very efficient manner in this model. Now, let's go back to the earlier time points. One of the first events that really follows this acute targeted injury, within 3 hours of inducing biliary cell death, we were able to see this very strong accumulation of CX3CR1 positive and also CCR2 positive monocyte-derived macrophages stuck to these biliary cells. And this is what you would see here with using this GFP reporter mice. And those cells were identified as monocyte-derived because they are positive for IBA1, but negative for CLEC4F. So the Kupffer cells are shown here in yellow, and these red cells are the monocyte-derived macrophages. We could quantify this. There was really a very significant increase in those macrophages, or monocyte-derived macrophages. And what was also interesting, if we deplete the macrophages in those mice, so we just injected clodronate-loaded liposomes, when we do that, we don't see a change in biliary cell injury, because the injury, I told you, is induced by this toxin, so it's a direct injury of the biliary cells. In this case, the monocyte macrophages do not seem to increase the injury, although they seem to somehow participate in the cholestasis. We did not investigate how, but this is the data we observed. And what was also striking to us was when we deplete the macrophages, we reduce the amount of repair of a biliary tree by seeing a reduced proliferation of the remaining biliary cells. So those macrophages were deemed unnecessary for the bile duct repair. So in short, in this project, we could evidence that acute biliary cell death induces portal inflammation, portal fibrogenesis, and I'm not showing you the mechanism, but we could also show that these macrophages were involved in the integrin beta-6 expression in the regenerating biliary cells. So this monocyte recruitment was really one of the first events happening within 3 hours after the targeted injury. And we could show that it participates in the initiation of a bile duct repair. So again, making a link with the previous project, this association between monocyte macrophages and biliary cells or ductular cells. Now, what we did not highlight in this paper was, of course, as part of this project, we did bulk sequencing, RNA sequencing. So to cut it short again, it was a microdissection of all biliary cells in those mouse livers, and we just ran a bulk sequencing. We could evidence a few mechanisms, including increased CCL2, so this chemokine attracting the monocytes, but it was not the only gene that was induced in those mice. And this is how I jump to the next project now. So in the next part of the talk, I really want to explain you this most recent project in which we investigated how the reactive cholangiocytes could drive macrophage reprogramming. So this project, I started it in Berlin now. We started by recovering this bulk RNA sequencing data from the targeted biliary cell injury model. So it's this data set. And we compared it to data sets of other models associated with, let's say, ductular reaction. So we just recovered publicly available data sets, and we looked at the genes that were common in those data sets. So the genes specifically expressed by biliary cells. So these are all data sets on purified or isolated biliary cells. And we came up with this short list of 5 markers, of which we decided to focus on ORM2 and ADAM11 because they were predicted to be extracellular released factors, so they could affect other cells in the surrounding of biliary cells. So this step was really fishing for some kind of biliary cell-derived signals that would happen during bile duct injury. So then we went on focusing on this ORM2 and ADAM11. We used a bit more of the publicly available transcriptomic data sets. So we recovered some mouse data sets, some human data sets, and in general, the signature of ORM2 was the most increased one in pretty much all mouse models of liver disease, whereas the ADAM11 was, let's say, not that significant or even decreased in some models. For the human disease, the ORM2 was increased in PSC and MASH, muscle D. For the cirrhotic livers, we saw a decrease, which was just as it is in bulk sequencing. So these were all bulk RNA sequencing data. But for the next part, we really wanted to go into single-cell data. So again, we used available single-cell data sets. We looked specifically at hepatocytes or biliary epithelial cells for ORM2 and ADAM11 expression. And what was quite surprising at this stage, so the blue reflects a decreased expression of ORM2, and ORM2 was decreased in hepatocytes, but increased in biliary cells. And this is what we observed as well by immunostaining on human liver samples from those chronically injured livers. So if you appreciate here in this, so it's again the same pattern of tissues. You have a healthy liver here. Here we have a muscle D end stage and PSC livers. The blue areas reflect the hepatocytes, and you would see the CK7 positive cells here. So these are ductular cells really massively accumulating through the liver. In the healthy liver, you would just see this small islet of cells, which are the healthy biliary cells. And in PSC, again, this ductular reaction here. And what was pretty clear to us, so this image appears rather red. That's just reflecting the very strong ORM2 expression in hepatocytes. And this is really going down upon injury in those livers. On the other hand, the expression was going to increase in CK7 positive cells. And this is what is shown on this graph. So in this graph, we have the cumulative ORM2 expression. So it's really trying to sum up all the signal we get from all hepatocytes versus summing up all the signal we get from CK7 positive cells. And the proportion of signal coming from hepatocytes is dropping when we go to increasing stages of muscle D and increasing in biliary cells. So for the next experiments, now we are switching to in vitro experimentation. So in this case, we used mouse primary liver cells mouse primary biliary epithelial cells isolated from mouse livers, or mouse hepatocytes, and we use them freshly. And in those cases, we applied different types of injury to the cells. So these are the controls here. Biliatrazone, a toxin-inducing cholangiocyte cell injury, or bile acid, CDCA, or APAP, acetaminophen. In those cases, we could always see an increased expression of ORM2 in primary biliary cells. For the hepatocytes, we applied acetaminophen, free fatty acid, biliatrazone, and we saw a decrease, at least in two of those conditions, in hepatocytes again. So we were confident we could indeed validate what we observed in the transcriptomic datasets. And this is also the data you would see here. So in this case, we performed immunocytochemistry, so staining on cell cultures. Injured biliary cells had increased signal for ORM2. Injured hepatocytes had reduced signal for ORM2. So in conclusion, this ORM2 gene expression is decreased in injured stressed hepatocytes and increased in injured reactive biliary cells. So just before I get the question, we don't know why it's decreased in hepatocytes. We asked our collaborators who are also working on ORM2. They don't know either, so that's a burning question if anyone wants to dig into this. But let's focus again on biliary cells. So first of all, what is ORM2? So ORM2 is part of the acute phase response proteins. It's found in plasma, and it's already known that its levels may increase during inflammation, autoimmune disorders, and cancer. It has been shown by the collaborators I just mentioned that it is indeed, it has been shown to improve hepatic steatosis by enhancing hepatocellular lipid metabolism and to directly promote macrophage-driven inflammation, thereby aggravating chronic arthritis in mice. But so far, for the liver macrophages, it was not known what it would be doing. So that's exactly what we went on to investigate. So we isolated, we freshly isolated primary liver macrophages from the mice, and we exposed them to recombinant ORM2, and then we perform bulk RNA sequencing. So what we observed was that indeed ORM2 was able to induce drastic transcriptomic changes in those macrophages. So in particular, it was inducing both the conventional M1 macrophages and the conventional M2 macrophages. So if you remember, I told you we don't like to say M1, M2 anymore. That's precisely because of this kind of transcriptomic profile. You may see macrophages with multiple profiles, typical profile expression. So again, we performed a similar analysis as the one I showed you before by comparing the gene signature of ORM2-treated macrophages with the signature of LPS, so lipopolysaccharides, or IL-4 treated macrophages. So LPS treated are the so-called M1, and IL-4 treated would be the so-called M2 macrophages. And we could evidence again that this was really a distinct gene signature in those ORM2 treated macrophages. And next, we went for the protein levels, and here we could see that by multiplex cytokine assay, that ORM2 was indeed able to induce the expression of all those cytokines: CCL2, TNF-alpha, IL-1 alpha and beta, IL-6, IL-10, and IL-23. So ORM2 is inducing a very specific macrophage phenotype, which we deemed distinct from that induced by LPS or IL-4. So again, ORM2 is expressed by biliary epithelial cells under stress conditions, and this ORM2 induces a specific macrophage phenotype. But then came, of course, the question, how can we directly evidence that this biliary cell-derived ORM2 is inducing a specific liver macrophage phenotype? I will give you some more technical notes now, on which we established a liver or biliary niche on a chip. So I'm sure most of you are aware there are a couple of in vitro models for liver diseases. It starts to be pretty widespread to use organoids. So you would use mouse or patient-derived cells that you grow as organoids. Or you could use precision-cut tissue sections. All those models have their advantages. In our case, we decided to go for a liver on a chip, which we exclusively based on primary cells. the main motivation for us was we needed a model to be immunocompetent. So this has been part of one of our publications as well. So originally, we designed this chip. So I will show it a bit bigger in the next slide. But you may imagine this is reflecting the schematic view of the liver sinusoids with the cell types that are present there. And we try to mimic that in vitro using our liver on a chip. And in this study in particular, we could evidence we can mimic acute drug-induced liver injury or acute free fatty acid-induced metabolism-related injuries. So this work was mostly performed by Hanyang Liu, who is currently experiencing NIH, which sounded like a good idea at first. And the work is continued now by Guillot. So if we zoom in a bit into this liver on a chip system, so it's the size of a glass for microscopy. You would have two independent units, and each of these units is made of two chambers separated by a membrane. This membrane being porous. So this is what you would see here. We have a porous membrane, which allows communication of fluid and we believe cells as well. And then we sit the cells on the two sides of this membrane. On one side, we would sit here the hepatic stellate cells or biliary cells. On the other side, Kupffer cells and endothelial cells. And the beauty of this chip system is also that we can perfuse. So it's a dynamic model with microfluidics. So in this case, we perfuse with circulating immune cells from the blood. So roughly, the protocols looks like this. We isolated the cells from the mice, directly seed them in the chip, or perfuse them through this lower chamber, which mimics the vascular layer. What we can get from this model, so just looking at the cells themselves, we can collect the membrane and perform gene expression analysis. We can also perform immunocytochemistry, and as well as multispectral flow cytometry on the circulating cells that are still remaining in the medium. In this medium, we can also measure metabolites by mass spectrometry, run the conventional lab analysis, so the enzyme activity, or measure the cytokines. In this case, flow cytometry, because we used a multiplex cytokine assay. And that will be an additional technical note for you to understand a bit. So in this case, what I will show you next, we decided to go for using biliary cells isolated from MDR2 knockout mice. So these MDR2 knockout mice spontaneously develop periportal inflammation and fibrosis and is regarded as a model of PSC. And they are marked by a really pronounced ductular reaction. So this is what you would see, the cyan cells here are marked for CK19. So we isolated those cells from the mice, grow them as organoids, and then use those organoids to seed them in the liver on a chip system. Why did we do this? I mean, first of all, we get more cells, they are reactive, so we consider them as a good model of ductular reaction. But we also came to realize they express higher levels of ORM2 compared to the wild-type cells. So let's go back to the ORM2 story now. So we seeded this biliary niche on a chip, which I just described to you, and we applied silencing RNA for ORM2. So we tried to silence ORM2 in these MDR2 knockout biliary cells. And when we did that, we could drastically reduce the recruitment of the circulating immune cells to the membrane in our liver on a chip. And what's more, when we silence ORM2, we could as well reduce the expression of inflammation and fibrogenesis-related genes. So that was a way for us to really highlight the biliary cell-derived ORM2 is driving directly inflammation in this system, at least. And next, we try to complexify a bit the model. So that was a very kind suggestion that was made to us by the reviewers, in which we combine hepatocytes and biliary epithelial cells. So now you can imagine the system with stellate cells, Kupffer cells, endothelial cells, hepatocytes, biliary cells, and we perfuse with circulating blood cells. So that did work. That's what you see here. So these blue spots here are actually the biliary cells that are still a bit forming these agglomerates because they derive from organoids. And we intentionally did not fully digest the organoids. So they are still making these blobs here. And then the hepatocytes are spread around, so they appear in green in this picture. And there you see the whole membrane in our biliary niche on the chip system. So just for you to appreciate the scale, it's a comfortable number of cells. And in this system, we could as well perform targeted silencing. So the way we did was we first seeded the cholangiocytes, organoids, transfected them with the siRNA against ORM2, and then apply the other cell types, and finally run the model. So that's what you could appreciate here. That's just looking at the cells here. So in green, we have the CK7 positive cells. Magenta is desmin for the HSCs. And you would see the other cells stained with DAPI around here. The CIC, so this is for circulating immune cells derived from the blood, which in this case we isolated from DS-red reporter mice. So that was a way for us to track the recruitment of those immune cells. And the hepatocytes, we just flag them based on the morphology of their nuclei. So they are very large nuclei. And in this model as well, we could evidence that when we silence ORM2, we could indeed reduce the recruitment of the LY6C high monocytes, which are typically regarded as the pro-inflammatory monocytes. So ORM2 knockdown in biliary cells, but not in hepatocytes. We did the same experiment, knocking down in hepatocytes. So only when we knock down in biliary cells, we prevent the accumulation of the LY6 high monocytes in this combined liver on a chip system. So coming to the conclusion, I'm really making this a short story, but we could evidence the injured biliary cells release ORM2 that through the ITPR2 receptor, so it's part of what I'm not telling you today, but you're welcome to check the paper, is inducing a release of cytosolic calcium. This engages signaling pathways within the macrophages, driving their switch towards this new phenotype of activation and active release of a lot of cytokines. And that, we believe, is part of how this crosstalk between biliary cells and macrophages is really driving at least some of the events associated with disease progression in the liver. So now I show you my conclusion. So I hope you understood and agree. Liver macrophages are very diverse. It's a highly dynamic immune cell compartment. And the cholangiocytes are not bystanders at all. So they do respond to injury. They do secrete a lot of factors that may influence directly their microenvironment. With this, I would like to thank you for your attention and, of course, all our collaborators and the team members that did all the work as well here. Thank you very much. From male mice, they were 12 weeks old, so not at all at the stage of cancer yet. And you're right, in vivo, there are differences. We assume it's not directly related to the biliary cells. So for us, it was important to make sure there is ongoing ductal reaction, and that's what was the case. I don't think there is one single factor that would be specific. What we call cholangiokine is anything released by cholangiocytes.
Okay.
So in this project, we did check the response of bone marrow-derived monocytes to ORM2, and we did not spot such a strong response. So it seemed that it was really the liver macrophages, so Kupffer cells, responding to ORM2 in this way. Again, we tried. we studied the different responses of monocyte-derived and Kupffer cells. In our hands, at least the naive, freshly isolated bone marrow-derived monocytes did not respond much to ORM2, whereas the liver macrophages did respond. Now, what I do not know for sure is if you were to isolate monocyte-derived macrophages already in the liver, already activated, they would certainly respond. That's what I would assume. I would like to say this is what I want to investigate in the follow-up projects. I don't think it is known already, honestly. Like I'm not aware of data sets that, I mean, the data sets would be available, the single-cell data sets, but I don't know if anyone did this analysis of comparing the genetic profiles between different etiologies specifically in ductular cells? I would love to have the answer, but I don't have it right now. Yes, so in terms of abundance, so when we seed the system, we just arbitrarily seed 1-to-1 ratios of all cell types so far. You are perfectly right. The next experiment would be to try different ratios, different activation status as well. Right now, we just take from healthy mice hepatic stellate cells. We were not able to see very strong fibrogenic response because I showed you the timeline, it's still a few days. And then I guess your question is also how much crosstalk there is between HSCs and biliary cells. I like to think there is a lot of crosstalk ongoing. We are going to investigate that again, but we did not do it yet. We focused on the macrophage biliary cell crosstalk. And also, thank you for pointing out, so we put the stellate cells on the side of biliary cells, precisely because when you do this immunostaining, you would see the stellate cells are very close to the biliary cells or ductular cells. So for us, it made total sense to just put them next to each other in the same compartment. It depends on your goal. We are having very active discussions with some people there, but I think it really depends on the goal. The transcriptomics have very high advantages. Of course, they are mostly unbiased, or you can run a couple of dozens of markers at once. I like very much the immunostaining-based approach because then you really see the proteins. And what to me is most important, especially with macrophages, is you preserve the shape of the cells. You can really see the membranes. And the macrophages, same as HSCs, they are not round cells. So I think in terms of segmentation of images, we are not there yet when we speak about spatial transcriptomics. So I would be a bit cautious with that. I mean, as long as you have a right antibody that can give you very specific signal, yes, sure. So this technique, again, we use off-the-shelf antibodies. So there is no specific conjugation, no specific formulation. You just use the antibodies you are used to use on your samples. If that works well, you can most likely integrate in this staining protocol we are using. And then if you get a nice image, you can run all types of analysis. Yeah. We checked just a few genes on all the cell types we seeded in the chip. So we applied ORM2 to each of those single cell types. We did not spot major response from anything else but liver macrophages. But it was really targeted PCR that we ran. I'm sure you would have specific markers you would be interested in.
I was not the reviewer, you see.
Yeah. You pointed out exactly what we are investigating long-term in our research projects. So in terms of transition of macrophages, I believe it's true, it's happening. The macrophages are highly plastics. They certainly switch phenotypes based on the direct clues they get from their close environment. At least here we did not look into that because we still had a definite set of markers. Again, if you have the appropriate antibodies, I'm sure you could try looking into this. Yet, these are static images, so you would probably need to go for other approaches, like intravital imaging, potentially. But yes, sure, I believe that's the way to go. And coming back to the first question, to me it's a very, philosophical question at this stage, and I actually asked it this morning as well during the talks. I'm not sure we can say ductular reaction is good or bad. It's like everything else, including inflammation. It's all about balance. So the organs respond this way to injury for an evolutionary reason, obviously. So there is a need for those cells to get activated, proliferate, and secrete what they secrete. Now, of course, when you have a disease progression, that means there is imbalance. So it's going towards the pathogenic side. I cannot tell you right now, I believe ductular cells are good or bad. I believe they do potentiate fibrosis and inflammation. But then I don't know if this inflammation is necessarily always bad either. You know, it's the same. It's again a balance between regenerative or pathogenic macrophages, for instance. So we have work to do for the next 20 years.
We'll be there.
Yeah, so for the second part, that's something we could consider doing, to just, as you said, isolate the cells, inject them in mice and see what's happening. For the first part, in terms of function, so we need to look into the proteins released by those macrophages. So I showed you there is quite a broad range of cytokines they release. So these are going to functionally affect the other immune cells or the other cells, like fibrogenic cells as well. And what we could evidence as well was the ORM2 treated macrophages had enhanced scavenging capacity, actually. So that would be as far as I know about the functional consequences of ORM2 treatment on macrophages. But thank you. This is also a very important question.
Thank you, Adrien. I think it's time to close this first part of the session. We're going to have a 10-minute break for a coffee, and then we'll be back sharp at 5:30 for Dr. Prakash Ramachandran talk. I really thank you again for your great talk. Thank you.
Thank you very much.